So now we'll be moving on and starting with proteins. ठीक है? Can you tell me where are proteins needed in our body? What's the importance of proteins that you know?
To make enzymes,
Good, exactly. They make enzymes. What else?
growth and repair of cells.
Good, good. So proteins are an extremely important class of macromolecular living organisms, and more than 50% of the dry mass of the most cell is protein. Proteins have many important functions. For example, all enzymes are protein. They make up the cell membranes. Hormones are even proteins such as insulin, glucagon. They also make up hemoglobin and myoglobin. Antibodies production for killing of microorganism and pathogen. Collagen protein, very important for tissues. Keratin protein for hair, nails, skin. Actin and myosin proteins in muscle contraction. And some storage products as well, for example, casein and milk. And despite the tremendous range of functions, all proteins are made from the same basic monomers. You always know that monomers make up what? Polymers. So here the polymers are the proteins which are made from their monomers, which are amino acids. Amino acids have a general structure, and there are how many different types of amino acids? 20. There are 20 different types of amino acids, and these are what? The monomers of proteins. We can see in this figure, this is a general structure of an amino acid. We always have a variable car group. Then we have a carbon attached to a carboxylic group and attached to an amine group. And on the third side, it is attached to the hydrogen. What does the variable group mean? Can you tell me? R group, what does it mean? That this group varies in different amino acids. For example, rest of the structure is going to be the same in all amino acids, but this R group can be CH3 as well. It can be C2HN. It can be anything. Okay, depending on 20 combinations of amino acid. There are total 20 different types. All of them would have the amine group, hydrogen, carboxylic, but this R group can be anything. It can be a variable group. This was a structure of a simple glycine, which is the simplest and the smallest amino acid. All amino acids have a central carbon atom, which is bonded to an amino group NH2 and a carboxylic group COOH. These two groups give the amino acids their name, and the third component that is always bonded to the carbon atom is a hydrogen atom. So the only way in which amino acids differ from each other is in the fourth group of atoms bonded to the central carbon. We call this the R group. Got it? This is a
Yeah.
general structure. Okay? Over here, we can see that the R group of glycine is what? Hydrogen. Rest of them are always the same. All three sides of carbon are same in all amino acid. Only this is going to vary. The R groups for the 20 different amino acids which occur in the proteins are also shown in a separate table, and you do not need to remember those, by the way. But you should know that all amino acids join together with the help of a peptide bond to make a polypeptide. A lot of amino acids are going to join together with the help of peptide bonds. This polypeptide now formed with many amino acids joined together is actually the protein. Figure 2.17 also shows how the two amino acids can join together. One of them loses a hydroxyl group from its carboxylic acid group, while the other loses a hydrogen atom from its amino group. This leaves a carbon atom of the first amino acid free to bond, and we call it a peptide bond. Let me show you how a peptide bond is made. Look at this. We have two amino acids, right? Lined in front of each other. This is the first amino acid and this is the second amino acid. So the first amino acid's carboxylic group is going to lose a OH. And the second amino acid's amine group is going to lose an H, which means water is going to be removed. And this carbon would directly bond with this nitrogen. This is known as a peptide bond. Look at the structure again and explain me if you have understood how two amino acids join together. Is it clear?
Yeah. So hydrogen and hydroxide becomes water, and then both form. Yeah. Understood.
जैसे remember the glycosidic bond forming between two glucose forming maltose? That is also the same ना कि carbon number 1 loses OH and the carbon number 4 of the other alpha glucose lost the H and they removed water. वो 1,4 glycosidic linkage था. This is known as a peptide bond. Got it?
Yeah.
And a lot of amino acids join together to form a dipeptide. Dipeptide means two amino acids join together. But a lot of amino acids join together. For example, in this structure, you can see so many amino acids join together. We call this a polypeptide chain. The new molecule is made up of two linked amino acids and it is called a dipeptide. A molecule made up of many amino acids linked together by peptide bonds is called a polypeptide. And a polypeptide is another example of a polymer and a macromolecule like a polysaccharide. A protein may just have one polypeptide chain, or it may have two or more chains as well. In living cells, protein synthesis takes place at ribosomes, and proteins can be broken down to amino acids by breaking the peptide bonds. Now, if you have to go back from protein to amino acid, you can carry out hydrolysis. Can you tell me what is hydrolysis?
Uh,
Adding water, breaking something by adding water. And how are they made? Condensation, by removing water. You forgot? What is the difference between condensation and hydrolysis? Tell me quick.
hydrolysis is when you break it down, and condensation is when you form it.
Condensation is when you're forming something by removing water. Hydrolysis is when you're breaking something and forming two products by the addition of water. It happens naturally in stomach as well. Even when in stomach हम proteins intake करते हैं. Now when we intake proteins, those proteins are broken down in our stomach as well by the help of proteases, and they are broken down into amino acids in our body. Then these amino acids are absorbed in our blood. So whatever we eat, whatever protein we eat is hydrolyzed in our stomach and small intestines. Clear?
Yep.
So can you tell me how many different types of amino acids are there?
20.
Good. And how are they joined together?
Wait, no, condensation.
Good.
And finally, we've got Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe the general structure of amino acids. You should then be able to describe how a peptide bond can be formed or broken. Now, proteins are among the most important molecules in biology. Proteins carry out a vast number of functions in living organisms, and all proteins are formed from amino acids. There are 20 different amino acids found in biology. So we're going to start by looking at the structure of amino acids. I'm showing you the general structure of an amino acid here. Now I should point out that this is not a specific amino acid. This is the general structure shared by all amino acids, and you definitely need to learn this structure. Now there are three main parts to an amino acid. We've got the amine group here and the carboxyl group here. These two groups are the same for every amino acid. And finally we've got the R group. Now the key idea you need to understand is that the R group is different for each of the 20 amino acids. I'm showing you two different amino acids here, and remember that you don't need to learn these specific examples. In the case of glycine, the R group is a hydrogen atom, and in the case of alanine, the R group is a carbon atom bonded to 3 hydrogen atoms. Now in the exam, you could be asked to name the elements present in proteins. Looking at the general structure of an amino acid, we can see that they contain the elements carbon, hydrogen, nitrogen, and oxygen. Some amino acids also contain the element sulfur. Coming up, we're going to look at how amino acid molecules can bond together by forming a peptide bond. Okay, I'm showing you here two amino acids side by side. In this case, these amino acids are both glycine, but this applies to any amino acids. Now, these two amino acids can react together and form a chemical bond, and I'm showing you that here. The bond formed is called a peptide bond, and you do need to learn that. As you can see, we've also formed a molecule of water, so this is an example of a condensation reaction, and we saw condensation reactions when we looked at polysaccharides. The molecule that we formed is called a dipeptide, as it contains two amino acids bonded together. This reaction takes place in ribosomes, which is where proteins are synthesized in cells. The reaction is catalyzed by a specific enzyme, and we'll look at that in a later video. Now, if we join 3 or more amino acids, then we make a polypeptide, and I'm showing you that here. As you can see, we've made one molecule of water for every peptide bond that we formed. Now, I should point out that polypeptides often consist of hundreds of amino acids joined together. Okay, so as we've seen, a peptide bond is formed in a condensation reaction, and a molecule of water is produced. Now, we can reverse this reaction and break the peptide bond, and to do that, we need to add back a molecule of water. This is called a hydrolysis reaction, and this reaction is carried out by protease enzymes in the digestive system. Okay, I want to end on one final point. As we've seen, when a large number of amino acids are joined together, then we call this a polypeptide. So what's the difference between a polypeptide and a protein? In order to be classed as a protein, a polypeptide has to fold into a complex three-dimensional shape. Once the polypeptide is folded into the correct shape, it can then carry out its function, for example as an enzyme or a hormone. And at this point, we'd refer to it as a protein molecule. Many proteins actually consist of several different polypeptides, forming a large and complex molecule. And proteins often contain other molecules, helping them to carry out their function. A good example of this is hemoglobin, which we look at in a later video. In the next video, we're going to start looking at the different levels of protein structure.
Is the basic structure of amino acids clear?
Yeah.
Good. So you should also know that when we talk about proteins, proteins are of total of 4 types. We know how proteins are formed. Simple hai, from amino acid monomers by the formation of peptide bonds. But proteins exist in 4 types. And those 4 types are a primary structure protein, a secondary structure protein, a tertiary structure protein, and a quaternary structure protein. If we talk about a polypeptide, what is just a polypeptide? It's a long chain of amino acids formed by condensation reactions between the individual acids. Because condensation reaction is carried out how? By the removal of water. We know that the carboxylic group removes the OH and the amine group loses the H. And this is how a peptide bond is formed. Primary structure is the very basic structure of proteins that we have already seen over here as well. It's a simple chain of molecules. Primary structure is the sequence of amino acids in a polypeptide or protein. And you can see that in a primary structure, a polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. The particular amino acid contained in the chain and the sequence in which they are joined is actually known as the primary structure of the protein. Simple arrangement. The simplest arrangement is known as what? The primary structure, when each amino acid is joined with the next amino acid by the help of peptide bond. No branches, no external, no globular structure, no jumbled up, no twisting, nothing. A straight, simple, coiled amino acid joint chain is known as a primary structure that you have already talked about in IGCSE and O levels as well. You can see this is a primary structure of ribonuclease, which is also found in pancreatic juice. So this is the most simple structure of a protein. Is it clear? What's the primary structure of protein?
Yeah.
So there are an enormous number of different possible primary structure, and a change in a single amino acid in chain made up of thousands may completely alter the properties of a polypeptide or protein as well. Even if one of these amino acid is changed or this is changed, it can actually alter the whole function. of the protein, because every gene codes for an amino acid. And when you change a gene sequence, it's basically exactly like you're doing mutation. You're changing the gene sequence. Here you are changing the amino acid sequence, which can completely alter the function of a protein as well. Next, we have the secondary structure, which I won't be starting today because I guess it's enough for today. We have three types of secondary structure. I'll just let you know. We have alpha helix, we have beta pleated sheets, and we have a triple helix. So this is a new concept that you'll be doing for the first time. Rest of it you can revise, and you can also send me the questions that you have solved for the carbohydrates topic.